So those are a few of the more important DNA tumor viruses that are known to cause neoplasms in humans. We know that something is special about those early genes that allows the cell to be transformed. But exactly how that happens had to wait until we understood how the RNA tumor viruses cause cancers. So let's now go on and look at the RNA tumor viruses. These are much less spectacular looking viruses compared to some of the beautiful viruses that are DNA tumor viruses, things like the adenovirus. I mean, they're just absolutely herpes virus. They're gorgeous looking things in an electron microscope. These are not particularly good looking viruses. They have, as I said, a RNA genome copied to DNA by reverse transcriptase. That genome now has to get into a chromosome, so the virus also codes for an enzyme called the integrase. Now, these are important virus-encoded enzymes, and they're of great importance because they are the enzymes that we can target for the Achilles heel of the virus, because this is doing something that we don't normally need to do. We don't need to copy RNA into DNA. We don't need to copy bits we don't need to take bits of DNA and stick them in our chromosomes. The virus does. And so it gives us an avenue of antiviral chemotherapy. And I'll talk to you about that later on in the week. These are virus-encoded enzymes, reverse transcriptase, integrase, and then there is this host-encoded enzyme that we use to make our messenger RNA, RNA polymerase II. And you can see here, there's a budding, these are all membrane-bound. Here it is budding out of the cell. And here is the mature form of the virus, nothing that you would find extremely exciting. And they're pretty small viruses. They normally have genomes of about 10,000 base pairs. And they have 3 genes. So if you're an RNA tumor virus, you can get away with just 3 genes to make more offspring. So they're pretty simple. One gene is called the GAG gene, and that stands for the group-specific antigens, and it really just codes for the internal protein. Another gene is called the envelope gene, and that codes for the envelope glycoprotein. There are usually more than one envelope glycoprotein. In fact, there are also many more internal proteins than one. So the initial primary translation product of these is cut up. A large polyprotein is cut up by a protease into the mature protein. And the polymerase, our third gene here, contains the enzyme, or the gene for the enzyme for that protease. So this is a virus-encoded protease that is used to cleave the initial internal proteins to their mature form. So you get one primary translation product from the GAG, one primary translation product from the pol, and then this protease cuts it up. Now, in the case of the envelope glycoprotein, that also has to be cut up because there's only one primary translation product here. That goes via the endoplasmic reticulum, and that's actually cut up by a Golgi body, host-provided protease. But these proteases, this protease function here is absolutely essential to get the single translation product of the pol and the GAG gene into the mature form in the virus, and therefore, if we hit it, this specific protease encoded by the virus is a good target, and it is the target, indeed, of the protease inhibitors that we'll be talking about later on in the week. So the pol gene, this third gene here, contains the enzyme genes, reverse transcriptase, also a ribonuclease H activity, the integrase, and the protease. Now, the RNA in the genome, there are 2 copies of it. It's a diploid virus. 2 copies are not complementary. There are 2 positive sense. That's the same sense as the messenger RNA. 2 positive sense RNAs there. Capped and polyadenylated, you might expect that. This is being produced by the host cell messenger RNA manufacturing system using RNA polymerase II. So you might expect the RNA to look rather like a messenger RNA, and that's exactly the case. Now, the virus is a positive sense. The virus genome is positive sense. But unfortunately, it's covered in protein in the virus, in the nuclear capsid. And as a result, even though it is message sense, it can't be read as messenger RNA once it goes into the cell. So that means that new messenger RNA has to be made when the virus infects the cell. However, to make new messenger RNA, you've got to go through the DNA form, right? You copy the DNA using reverse transcriptase into DNA, the RNA using reverse transcriptase into DNA, and then you use polymerase II to make more messenger RNA. So this copying of the positive sense messenger of genomic RNA into messenger RNA requires the DNA form. The DNA form requires reverse transcriptase. Reverse transcriptase is not in the cell because it's not a normal cell function. It has to be provided by the virus. But viral proteins can't be made unless you have the action of reverse transcriptase. Therefore, the virus has to do something. It has to bring molecules of reverse transcriptase into the cell with it so that it can copy that RNA into DNA. The DNA can be copied to RNA, and then the RNA can be translated into more reverse transcriptase and more other proteins. So each particle of a retrovirus will contain up to about 10 reverse transcriptase molecules, protein molecules, to get the thing going once it infects a cell. And here's a diagram of the virus. Although there's only 3 genes, clearly a lot more proteins, the primary translation products are cut up and inserted at their correct place in the virus. This, in fact, is HIV. So which RNA tumor viruses cause human cancers? Actually, we don't know of many that do. There are 2 groups of retroviruses that are reported. There's actually 3 groups of retroviruses, but one of them we don't really bother to think about. There are 2 groups of important retroviruses, the oncovirinae, and these do cause human neoplasms, and the lentivirinae, which until the 1980s was considered to be primarily a problem in veterinary studies because the archetypal virus was Visna, which is a virus that infects sheep. These have a long latent period. They cause a chronic progressive disease, and that Visna is a chronic progressive disease of sheep. But in the 1980s, HIV, a member of the lentiviruses, was discovered, and so now this has become obviously very important for humans. So which retroviruses cause human cancers? There are really only 2 that are known to do this. In the 1980s, there was a big search for the human cancer retroviruses by a lot of people who thought that there were going to be a lot of them that were discovered, and there was a big race to find the first human cancer-causing retrovirus, but as I said, only 2. They both involve neoplasms of lymphocytes. First one to be discovered was human T-cell lymphotropic virus 1, HTLV1. It causes some not all that common lymphomas. It's adult T-cell leukemia, Sézary T-cell leukemia. They're the same thing. Sézary was the French physician who discovered this disease. It's got a weird distribution throughout the world. Africa, the Caribbean, some Japanese islands, and particularly in certain parts of northwestern South America. It was originally thought that maybe it was the Japanese or the people who moved from Japan into South America brought the disease with them. However, there's now been the suggestion that evidence that this disease was actually found in people before the Japanese people moved to South America. But anyway, it's an odd distribution around the world, but this Sézary T-cell leukemia is caused by HTLV1, and about 1 to 4% of infected people go down with this problem. It also causes a disease called tropical spastic paraparesis, and that involves a myelopathy of the gray and white matter of the spinal cord. And again, about 4 or so % of people infected by HTLV will go down with that. Second one, HTLV2. It causes hairy cell leukemia. This is found in Native American populations primarily in this country, in people like Navajo and Seminole Indians, and it's also found in Native American populations in Central and South America. Very high seroprevalence, particularly women over 50. Up to half of women over 50 show antibodies against HTLV2. And the reason it's called hairy cell leukemia is because the cells have all these little projections all over them, and so they look rather hairy. And there's always the possibility, as we shall see next time, that HIV might be a third retrovirus that can cause cancer. Now, we know that the neoplasms associated with HIV-infected patients, things like Castleman's disease, Kaposi's sarcoma, are not caused by the HIV virus itself. HIV is a cofactor. The immune suppression caused by the HIV is what leads to the emergence of things like human herpes virus 8. But nevertheless, HIV is a retrovirus and has all the capacities, as we shall see, to cause cancers. And that's been a great problem for the development of anti-HIV vaccines. Because if we develop an anti-HIV vaccine that is an attenuated vaccine, we may be substituting a virus that can cause immune deficiency with a virus that can cause cancer. So there's been a lot of reluctance to think about vaccines against HIV, as we'll see again later on, in regard to attenuated vaccines, because of the possibility that HIV may also be a cancer-causing retrovirus. So let's look a little bit at their life history. They bind to a surface receptor. They usually bind to very specific receptors. HIV binds, as you know, to the CD4 antigen on T4 lymphocytes. In many cases, endocytosis follows. In some cases, such as HIV, the virus membrane can actually fuse with the cell surface membrane, leading to syncytial formation. But either way, endocytosis or fusion of the plasma membrane, you get the nuclear capsid into the cytoplasm. And on that nuclear capsid, there are targeting signals which will take the nuclear capsid over to the nucleus. And it is there that reverse transcriptase starts to produce this form of the virus that can integrate into the host cell chromosome. So here's the parental RNA in the virus. Reverse transcriptase comes along and forms a hybrid. That ribonuclease H activity of reverse transcriptase now gets rid of the RNA here, and you get reverse transcriptase now acting as a DNA-dependent DNA polymerase, forming a double-stranded DNA. That circularizes. The virus-encoded integrase inserts it into the chromosome. The host DNA polymerase copies that as the cell chromosomes are copied. So the virus gets perpetuated in the chromosomes, and that's a major problem with long-lived cells which are HIV-infected, as again we'll see later on. That DNA is now a gene. It gets transcribed by the host RNA polymerase II into the genome. Host splicing enzymes make messenger RNA, and protein is formed. Now, there's a big drawback to this. If you were designing yourself to be a virus, this kind of virus, you wouldn't do this, because there is a major problem for a retrovirus in pursuing this kind of lifestyle, of taking a genomic RNA, copying it into a DNA copy, and then copying that DNA copy back to messenger RNA. That is done by reverse transcriptase, as I've told you several times. That is done by the host RNA polymerase II. The problem is another problem of using host polymerase II. I've told you one problem for the lifestyle of retroviruses in using this host cell enzyme, polymerase II, that is, it doesn't have a proofreading capacity. And so as a result of that, you get enormous mutation rates in retroviruses. And that, of course, is a problem for vaccines. But there's another problem in using an enzyme that was designed to copy DNA in a gene into a messenger RNA. When Pol2, mRNA, RNA polymerase II, copies a gene, it doesn't copy the whole gene. It doesn't need to. There are parts of a gene that have nothing to do with the information that will get transcribed into messenger RNA and translated into protein. There are parts of a gene which are designed or used to control that process, but the information is not perpetuated into the messenger RNA and translated into protein. These are things like the enhancers and the promoters and the initiation site and the termination site. All those are part of a regular gene, but they don't turn up in the messenger RNA because to make a protein, they're not needed. So here again is the problem faced if you use this kind of lifestyle. Here's your RNA, genomic RNA, coming in to the infected cell, and you've got an RNA-dependent DNA polymerase, and all DNA polymerases need primers, and in the case of the retrovirus, that's a transfer RNA that the virus picked up in the previous cell that it infected. So it always carries along with it a few of the old host cells' transfer RNAs to use as its primer. And then the reverse transcriptase copies that. Okay, we've now got a strand of DNA. Reverse transcriptase now copies that back, as we've seen, to form double-stranded DNA. Now what happens? You've got an RNase initiation site that is never at the end of the DNA in the gene. Upstream from here, you've got the promoters. So RNA polymerase 2 comes along, and it says, here's the gene. Okay, I'll land on a promoter there, and I'll scan around, and I'll find the downstream initiation site, and I'll set up shop there, and I'll copy it till I find the termination site, which is ahead of all those enhancers and other control regions that are downstream. And as a result, we copy a shorter form of the gene. So, problem. The virus needs, when it forms its DNA form, needs to have those control sequences in, but they're not in the RNA, which came from a previously infected cell and was made by RNA polymerase 2. How does the virus overcome this problem of not being able to copy the upstream sequences, the promoters, the enhancers, the initiation sites, poly-A sites, termination sites, and all those other good things that are parts of all genes but are never copied into messenger RNA? So, maybe the virus could integrate itself somewhere in the chromosome so that it was downstream from an RNA initiation site, it was downstream from some enhancers, etc., downstream from promoters, upstream from enhancers, etc. That would be a pretty tall order to do, to get in there, but on the other hand, it might be possible. Alternatively, the virus could carry in its RNA form those promoters, enhancers, and all the other good stuff that a gene needs, but then it said not copied. Well, the clue to how it does it, and it's absolutely, it's amazing how the virus does this. I almost think that these things think for themselves. The clue to how this happens, which is also very important to the biology of tumor formation, comes from a subtle difference, or maybe not so subtle difference, in the structure of the RNA and the DNA form. So let's just look at the structure of the RNA form here. Here are the three genes, GAG, POL, and N, that I told you about. Is all that's necessary to make a new retrovirus. Upstream, there are some regions that are not used to make viral protein. There are two repeat regions, one here and one here, at either end. They're repeat regions. They're quite common in virus biology, as you've heard. And then there's two unique regions here. There's a thing called the U5 here, near the 5' end. And here, near the 3' end, there's a region called the U3 region. So these are the repeats here. And then before you get to the first protein coding gene, you've got a U5 region. And here, after the envelope gene, you've got the U3 region. And on the DNA form, there's a difference. You've got what are called long terminal repeats. And here, what you can see has happened is that this U3 region here has appeared here as well. And this U5 region has appeared over here as well. So now we've got U3RU5, U3RU5, called long terminal repeats in the DNA form. And that's not something we see in the RNA form. And this is what's happened. During the copying by reverse transcriptase, by an increasingly well understood but still not completely understood mechanism that involves circularization of the virus, etc. During the copying of the RNA form, that U3 region gets duplicated and put over here. And then you get the copying of the regular part of the viral RNA, and then that U5 region gets copied and put over there to form these long terminal repeats. Now, it so happens that there was an RNA initiation site and some promoters in this U3 region, not where it ought to be, but it's just been carried along here. And when that has been put over here, the U3 site gets put over to here, and it turns out that over here there is an RNA initiation site when it's in the DNA form. And so we've got an RNA initiation site there. In the region up here, we've got a termination site, and we've got enhancers. Also in this site, there are promoters. And so we get an RNA termination site right here. So the virus has got cassettes of information for promoters and all the other things actually inside the RNA form that then during reverse transcription get put into the right place. So that you've got a promoter here. Polymerase II can find that promoter, move along to the RNA initiation site there, copy the RNA into a DNA into an RNA form, terminating here, and you've got back to what you started with. Now, isn't that just absolutely fantastic?
What would stop it from stopping at the first?
That is a very good question, to which we do have an answer. There's a termination site, as you say, here, that in the U5 that gets transposed over here. That U5 there should contain the same termination site, and it does. But just in the beginning of the gag protein here is a sequence which causes the RNA to, or the DNA, to be to assume a secondary conformation that actually suppresses the first of those termination sites. So yes, the RNA should be started out here and should go down there to the termination site there, but it doesn't. It's actually read through in most cases because of a suppression sequence which is actually in the first gene, the beginning of the first gene. It's a puzzle that a lot of people have asked about. So this means that retroviruses then, if they're carrying their promoters in that little cassette internally in the RNA form, can only have one promoter. It's in the U3 that gets shoved over here. So that means there can only be one RNA read off the gene. Okay? Because there's only one promoter site here, one RNA initiation site that was created in the DNA form at the right place. So only one long RNA can be made from the DNA form. Therefore, you've got to process that RNA, and for that you are going to usurp the RNA processing enzymes in the nucleus of your host cell, the host enzymes. And this explains why the RNA must be positive strand. Because the RNA has to function both as a genome in its full-sized form and also as a messenger RNA, either in its full-sized form or in a spliced form. And in fact, what happens is that genomic RNA, which will be processed, which will be packaged rather into the new viral particles, also acts as a messenger RNA for GAG and POL. Host cell splicing of that messenger RNA cuts out the GAG and POL, gives you the end gene, and then that is translated into the envelope glycoprotein. And I see the time now has come round to 12:50, or 11:50 rather, so it's lunchtime. I wish you a good lunch, and I'll see you at a horribly early time tomorrow morning.